Yeah, hi, this is Mathias Lichterfeld here. Thanks a lot for inviting me for this presentation. And I also wanted to say thanks to all of you who are able to join this meeting and Zoom in. So I'm going to talk a little bit about recent progress in next generation sequencing technologies and what we can do to better understand HIV reservoirs and finding a cure for HIV. And just to briefly give some brief definitions here. So by HIV reservoir cells, we essentially refer to all the HIV-infected cells that are persisting during effective antiretroviral treatment. And those are the cells that are fueling rebound viremia in case of treatment interruption. So this slide just summarizes a few details of what we know so far about HIV reservoir cells. So we certainly know that these are very small numbers of cells. They represent about 1 to 10 cells in a million CD4 cells, and they are able to persist really forever, for the entire lifetime of an infected individual. And that's due to integration of HIV into the host chromosome. So HIV becomes part of the human genome. And what we also know is that these cells are not susceptible to the type of antiretroviral treatment that is currently available. And if we had an opportunity to get rid of these cells by some type of therapeutic intervention, this would probably result in a sterilizing cure of HIV. So analyzing these reservoir cells is quite difficult, actually, for a number of different reasons. One of the reasons is that we have to make very fine distinctions between intact HIV proviruses and defective proviruses. So here on this slide, you see an intact provirus, but in fact, the vast majority of proviruses are defective. The intact proviruses can build functional viruses, but the defective proviruses, they are just sitting in the chromosomal DNA and cannot produce functional viruses. And these defective proviruses really occur due to defects in HIV reverse transcription. So the viral reverse transcriptase that is doing this is making a lot of mistakes, and that's why the vast majority of HIV proviruses that are ending up in the chromosomes are defective. So how can we distinguish intact and defective proviruses? That's really where we can take advantage of next generation sequencing technologies. And one of these technologies is shown on this slide here. It's an approach that we call FLIP-seq, which stands for full-length individual proviral sequencing. And it works in a way that patient-derived CD4 cells are diluted to single HIV genomes in this limiting dilution process. And then we are using a full-length PCR that is amplifying the entire genome, and then each individual genome is sequenced on the Illumina sequencing platform. And subsequently, all the sequencing data that we're getting is entered into this sort of decision tree type of software that you can see here. So it's a computational process that identifies different types of defects in genomes, and we can sometimes identify large deletions, we can find hypermutations, we can find premature stop codons, for example, but we also find a small subset of HIV genomes that tend to be intact and don't show any type of defects. And those are the sequences that we are ultimately interested in. And using this computational approach here, we can very effectively distinguish intact and defective proviruses. And just to give you an example of how this looks like, here we are showing data from one particular study person. Each of these horizontal lines here is one particular sequence that was isolated and sequenced. And you can see here, based on this color coding, the vast majority of these sequences that we picked up here had some type of defects. Some were hypermutated, some had large deletions, some had premature stop codons. And there's only a very small subset of these sequences that actually are genome intact and encode for replication competent HIV. In a subsequent analysis step, then we have started to go a little bit further and combine this assay with a platform to identify the chromosomal location of these intact proviruses, which we believe is very important because it makes a huge difference at what position in the genome HIV is sitting in. So in order to do this, we developed this approach here that's called MIPSeq. It stands for Matched Integration Site and Proviral Sequencing. Again, the workflow here is that individual proviruses are diluted to single genomes, but instead of using these single genomes then for direct amplification of HIV, we are doing a whole genome amplification step first. And that whole genome amplification, which we are using, it's called multiple displacement amplification, is designed to increase each proviral genome by about 1,000 to 10,000 fold. So it's creating identical copies of that individual provirus. And then we can split each multiple displacement amplification reaction in two different components. One of them is being used for HIV amplification, and one of them is used for integration site analysis. And using this approach, we can identify the entire virus sequence and the corresponding integration site in the human genome. This slide shows an example for these types of studies. So we are seeing here on the circus plot all the different chromosomes that are listed, and we are showing for one patient the different types of proviruses that were isolated and their relative location in the human genome at different positions. And then we can make a lot of distinctions here. We can certainly distinguish the intact and the defective proviruses based on color coding. They're indicated as red and blue here. We can certainly distinguish proviruses that are integrated into the same direction as the host gene or in the opposite direction of the host gene. And we can also find certain HIV proviruses that are integrated in non-genic regions, so in non-coding parts of the genomes. So this gives us a lot of information to better understand where HIV is sitting and specifically what kind of viruses are sitting at what positions in the genome. So this again here is an example of this. We ended up doing this assay in 3 different patients in this context here. And you can see here these phylogenetic trees that are just reflecting intact proviruses that were isolated from these different patients. And the chromosomal location is indicated for each of these proviruses. And what you can see here is there's a lot of diversity, lots of different types of proviruses have been identified, and they are all sitting at different parts in the human genome. So we use this technology then to move on and look into elite controllers. And most of you know this, probably elite controllers are HIV-infected patients that manage to have undetectable levels of HIV viremia in the absence of treatment. And in a way, you could argue that these patients are the closest possible approximation to a natural cure of HIV that is possible. So we use these technologies that I just talked about to better analyze the reservoir cells in elite controllers. And here you can see, what you can see here is that we analyzed intact proviruses in a large cohort of elite controllers. So we looked into 64 different elite controllers, and we also had a comparison cohort of 41 ART-treated patients. And each dot here represents one study person. And you can see that in elite controllers, on this population level, the proportion of intact proviruses that we can detect is significantly lower compared to ART-treated patients. But at the same time, you can also see that there's a lot of diversity in this ART-treated patient cohort. Some patients actually have very high levels of intact proviruses, and some patients have very low levels of intact proviruses. So that gets us to the question, where is HIV located in these elite controllers? Where is it located in the human genome? And we are first focusing on one patient that is highlighted here by this arrow. So it's relatively representative of this entire population of elite controllers that we analyzed. And here you can see a phylogenetic tree that shows all the different sequences that were isolated from this patient. And you can see here is one clone of sequences. So this is highlighted here in this type of cluster. These are all sequences isolated from this patient, and they are all 100% identical. And we believe those are sequences that are deriving from cells that are actively proliferating, clonally proliferating in patients. And you can see that this phylogenetic tree here is very limited. It's really essentially just this one clone that is present in this patient. And then we can see where is it located in the human genome, and we find that it is integrated in chromosome 15 in the center of the chromosome, so what we're calling that centromeric region here. And that is actually very unusual. So this centromeric region, it's also called satellite DNA, and we find that this location is very atypical in non-elite controller patients. In the vast majority of ART-treated patients, HIV is actively avoiding this region, and it's not integrating in this region. This is a region that doesn't encode for any genes. That's mostly structural, non-coding DNA. It's also been called a gene desert. And once HIV is sitting at this particular location, it seems like this is sort of a dead end. HIV can really not be transcribed when it is sitting in this very unusual location. And that's why this particular location has also been referred to as a block and lock location. So once HIV is sitting there, it cannot be expressed anymore. I'm showing one more patient here. So this is a patient that had relatively high numbers of intact proviruses. It's shown up here in this red arrow. And here again is a phylogenetic tree that shows all intact proviruses that were isolated from this patient. And again, you can see here these clusters of intact proviruses that are 100% identical, which derive from clonally expanded reservoir cells. And now we analyze the integration sites of these different types of intact proviruses. And you can see here for 3 of these intact proviruses, we find that they are again located in that centromeric satellite DNA. We have 2 more that are also in centromeric DNA, just in immediate proximity to the satellite DNA. And only 1 of these intact proviruses is actually located in a genic region. So we see here, this is a very atypical profile of integration sites in elite controllers. Most of these intact proviruses are actually located in centromeric satellite DNA in regions that are very unusual in ART-treated patients and associated with transcriptional repression. I briefly wanted to talk a little bit more about how this relates to a cure for HIV. Most of you know about this patient, Tim Brown, who was the first patient who was actually cured of HIV by getting a bone marrow transplant that was positive for this Delta 32 variation, which confers resistance to CCR5 tropic HIV infection. You may have seen the recent news that he actually had a recurrent leukemia, which was the original reason for getting a bone marrow transplant, and he just died relatively recently. But he has sort of been a model and a paradigm for how a cure for HIV would possibly be inducible. Our question was, to what extent are these elite controllers, to what extent do they have achieved a sterilizing cure? And as I've seen, as I've shown you earlier in some of the slides, in the vast majority of elite controllers, we can really detect intact proviruses. So they don't have a sterilizing cure. But some elite controllers may actually have a sterilizing cure. So that's a little bit what we wanted to investigate. And we focused on this one patient here, who we call the San Francisco patient. So this was a patient who was diagnosed back in 1992, was treatment naive at this time, and then maintained undetectable HIV viremia for 24 years in the absence of treatment. And during this period of time, this person had 39 different viral load tests, and all of them were negative except for 1 test back in 2000, when a total of 93 copies were isolated. So we analyzed this patient here in detail, and we ended up analyzing massive amounts of cells from that patient. It was 1.5 billion PBMC that were subjected to this FLIP-seq approach that I had talked about earlier, so that single genome amplification process. And out of these 1.5 billion PBMC, we failed to identify a single intact provirus. We found 4 hypermutated sequences, and we found 15 sequences with large deletions. All of these are defective proviruses. And it's clearly indicating this, that this patient was infected with HIV at one point in the past. But we have failed to identify intact proviruses out of 1.5 billion cells. We also did additional studies here. So we did a QVOR, which is a quantitative viral outgrowth assay. A total of 340 million resting CD4 cells were isolated and subjected to that assay. in one experiment, and there was a second experiment when 41 million total CD4 cells were isolated and subjected to this experiment. And in both cases, we failed to identify any replication competent virus. And at the same time, we also conducted this IPDA assay. It stands for the intact proviral DNA assay. A total of 14 million cells were analyzed in this particular system, which is basically a PCR assay. And again, we failed to identify a single intact provirus using this particular technology. And these types of assays here were conducted in collaboration with Bob Silicano's group. So essentially what this means is that in this particular patient, despite analyzing massive amounts of cells using a variety of different types of assays, we have failed to identify a single intact provirus. And certainly, it doesn't prove that this person has achieved a sterilizing cure, but it very strongly suggests that this person actually is cured and should be regarded as a cured patient. So just to summarize the major findings of this talk, what I've shown you is that there is one subgroup of elite controllers who have extremely low levels of intact proviruses, and one particular person who may actually have achieved a spontaneous cure, a spontaneous sterilizing cure of HIV. But there's also a second subgroup of elite controllers that have relatively high numbers of intact proviruses, which may sound paradoxical to start with, but what we show is that's really about the positioning and the location of these proviruses. And what we have seen here using the new types of technologies that we have developed is that intact proviruses in elite controllers are really located in highly distinct positions of the human genome that are associated with heterochromatin features and are essentially repressed and not transcriptionally active. And we believe that these particular locations in the genome play an important role in maintaining drug-free control of HIV for extended periods of time. I just wanted to acknowledge a number of people who have contributed to our work here. They are listed on this slide, and I also want to thank the support from Harvard University for this work.